Hi, we are Tu and Harry Hara from the University of Maryland. This is a joint work with Marvin, who was an undergraduate researcher in our lab, and Professor Mihai Pop. Today, we are presenting SCRAPT, an iterative algorithm to cluster large 16S rRNA gene datasets. Microbes are everywhere and have co-evolved with human beings. Taxonomic characterization of microbiota is an important task in microbiome analysis. Many studies have relied upon marker genes such as 16S rRNA to understand the microbiome composition. A typical analysis pipeline begins with sample collection and sequencing. Upon extracting the DNA, we perform PCR amplification process to identify sequences markers such as 16S rRNA gene using specialized oligonucleotide probes. We then rely on computational algorithm to cluster the 16S rRNA gene markers into OTU operational taxonomic units, or ASV, applicant sequencing variants. Those OTUs are then used to perform downstream analysis such as taxonomic annotation, differential abundance testing, etc. Several tools such as DNAclass, uClass, uSearch, and CityHit have been proposed in the past to perform OTU clustering. All these methods take as input a similarity threshold and build clusters that maintain the similarity using a graded clustering approach. These tools maintain a schedule of sequences sorted in decreasing order of their length and iteratively assign sequences to clusters by comparing them to existing set of representatives. Another notable clustering approach is data2, which tries to identify a set of sequences called applicant sequence variants and their abundance in a sample that best explain all the input sequences under a model of sequence arrow and evolution. However, they either produce fragmented clusters or spend a lot of time in detecting small clusters or singletons that are eventually discarded due to the absence of any statistically significant signal. Our experiments also showed that the existing methods produce fragmented clusters, emphasizing the importance of picking the correct centroid. Here, we want to give an overview of the iterative clustering framework. Given a set of 16S rRNA gene sequences, we deduplicate it and maintain a dictionary of their respective counts. We start by sampling the unclustered sequences uniformly. We then cluster the subsample using DNAclass, which gives us non-singleton clusters and singletons. Baiting is a process of retrieving the other sequences in the full dataset that belong to the corresponding cluster with a certain similarity threshold. We use DNAclass to bait the centroid of the non-singleton clusters on the unclustered sequences from the entire dataset. Due to the uniform sampling we perform, the initial centroid obtained from the subsample may not be the best representative. Therefore, we shift the centroid of the non-singleton clusters to the mode of the clusters. The mode of a cluster is a sequence with the largest number of identical copies in the dataset. Then we bait using the new centroids on the entire dataset. We remove all the clustered sequences and repeat the process on the remaining unclustered sequences in the dataset. In this algorithm, we use DNAclass as the clustering method, but it can be replaced by any other methods that has appropriate clustering and baiting functionality. For example, DNAclass has both such functionality and therefore can be replaced for DNAclass in our algorithm. As mentioned earlier, our iterative algorithm relies on a sampling step. We make a probabilistic argument on why a naive sampling, that is, a fixed sampling rate for all iteration, will converge soon without discovering all clusters. We show that naive sampling is likely to bias towards large clusters and misses small clusters. We see an exponential drop in the probability of missing a cluster as its size increases. We show that the probability of a cluster being ignored begins to drop with increasing sampling rate. This means in order to recover small clusters, the sampling rate has to be large. One thing that the probabilistic analysis failed to convey is how the distribution of clusters changes as we shift from one iteration to another. This is because the distribution of the cluster sizes is not known in advance. Therefore, we stimulated cluster sizes from different distribution, and we studied the entropy of the cluster discovered as the algorithm proceeds. We see that as the number of unclustered sequences drop, the entropy of the clusters discovered in each iteration also drops, indicating the remaining unclustered sequences comprise of a fairly uniform set of small clusters. The naive sampling is expected to identify and remove large clusters in the initial few iterations, causing the data to be depleted of large clusters and thereby increasing the odds of singletons in later iteration. Therefore, to identify all clusters of a particular size of interest, we propose an adaptive sampling-based clustering algorithm. We have a few observations. First, large clusters have high probability of being clustered in the first few iterations. Second, our sampling rate alpha can be viewed as the total number of sequences we draw over the number of non-singletons plus the constant C, which is the total number of singletons in the original dataset. As more and more sequences from the large clusters are removed from the set of remaining sequences, the total number of non-singletons decreases. Therefore, the chances of forming new non-singleton clusters with a constant sampling rate alpha decreases, since the majority of sequences in the late iterations are singletons. Here, we introduce a strategy called adaptive sampling that helps speed up the algorithm in early stages and helps to form clusters in late stages of the algorithm. We increase the sampling rate by a fraction of the relative difference in number of sequences clustered in the current iteration to the previous iteration. Suppose the total number of sequences clustered in the iteration I is d sub of I. We adjust the sampling rate alpha proportional to absolute d sub of I minus 1 over d sub of I times the rate constant sigma. We implement the iterative clustering with adaptive sampling and mode shifting through SCRAPT. To evaluate SCRAPT, we rely on two datasets of different sizes. The first dataset that we refer to as lupus microbiome dataset in the remainder of this talk contains about 6 million rays and resulted in about 900,000 rays after deduplication. The second dataset is a soil microbiome dataset sequenced as a part of the Earth Microbiome Project. This is a deeply sequenced dataset and had about 98 million rays, and this resulted in about 16 million rays after deduplication. To illustrate the benefits of running SCRAPT, we benchmark the total number of sequences that belong to non-singleton clusters across all methods we test, the extent of fragmentation in the clusters and their respective runtime. To compute the extent of fragmentation, we order all the clusters in decreasing order of their sizes, and for every size x, we measure the total number of sequences contained in clusters of size x or greater. This mirrors the approach used to evaluate the contiguity of genome assembly. We draw your attention to the figure. We compare two cluster method A and B. The method A is said to be less fragmented than method B. Higher the curve, the lower extent of fragmentation. To study the performance of SCRAPT, we compare SCRAPT against other variants of iterative clustering, namely naive sampling with and without mode shifting, and the adaptive sampling with without mode shifting. We also compare against other popular ODU picking methods, namely DNA class, CD-HIT, U-class, and Data2.
In this figure, we show the cumulative number of sequences clustered by different variants of the iterative clustering. We compare the performance of SCRAPT, which uses an adaptive sampling and mode shifting, against clustering variants that use a naive sampling with mode shifting, a naive sampling without mode shifting, and an adaptive sampling without mode shifting. As seen from the figures in the left panel, we see that the methods that use adaptive sampling converges independent of the choice of initial sampling rate, while naive sampling, as seen from the figures in the right panel, saturates early on, especially for small initial sampling rates. It's key for the performance of SCRAPT to have a small initial sampling rate to maximize performance gains. These curves do not convey the importance of center selection. We show that the choice of centroids determine the clustering qualities in the later slides. We computed the extent of fragmentation for the lupus and soil microbiome datasets for different values of initial sampling rate alpha. We see from the curves in the top panel that the trends of fragmentation are alike for the lupus microbiome dataset for all values of alpha. As seen from the curves for the soil microbiome dataset, we see that the size of the largest clusters seem to vary for different initial sampling rates. We attribute this variation to the relative noise content in the dataset. However, we show in the next slide that this still outperforms all the other clustering methods. We also show the cumulative number of sequences clustered versus the time taken per iteration on the bottom left figure. We plot the number of sequences clustered and the time taken by DNA cluster when run standalone to provide a frame of reference. We see that SCRAPT identified most of the clusters only in a fraction of the time taken by DNA cluster. We finally plot the distribution of cluster sizes identified in each iteration in the soil microbiome dataset, with the largest cluster highlighted in red. As seen in the figure on the bottom right, the largest clusters in the dataset are rapidly detected in the first few iterations, after which the clustering process reaches a plateau, during which, in each iteration, it identifies a subset of the small clusters in the dataset. Depending on the application, the clustering process may thus be terminated as soon as the relevant clusters have been identified. We compare SCRAPT and its variants with popular OTU clustering tools including DNAclust, cdHit, and uclust for the same similarity threshold in terms of their runtimes and fragmentation measure for the lupus and soil microbiome datasets. It seems from the plots on the left panel that for lupus microbiome dataset and soil dataset, both SCRAPT and naive sampling with mode shifting perform similarly on the fragmentation measure, while they outperform the other variants and DNAclust, cdHit, and uclust. We've seen from previous slides that naive sampling and mode shifting can miss out in smaller clusters. This highlights the importance of picking the right centroids, and SCRAPT's ability to shift to the right centroids results in good quality clusters. In general, SCRAPT also runs faster than other popular OTU clustering methods, including DNAclust, cdHit, uclust, and DADA2 in both the datasets. From the two plots on the right panel, we see that the runtime of SCRAPT is independent of the similarity threshold, while all the other tools took times proportional to the stringency of similarity thresholds. One of the reasons for SCRAPT's efficiency is that the baiting can be done in parallel efficiently. We also note that we do not show the results of uclust for the soil dataset, as the open source version of uclust failed to produce outputs. We assess how similar the output of SCRAPT is to DADA2's, which is currently the widely accepted standard for 16S rRNA gene sequence analysis. DADA2 takes the entire data set as an input and outputs the top denoise sample sequences along with the number of sequences within the ASV. For each denoise sequence identified by DADA2, we found the largest SCRAPT centroid that had at least 99% similarity with the former. We then computed the number of sequences within the corresponding SCRAPT cluster for each ASV associated with DADA2. It is evident from the plot that the sizes of the clusters discovered by both the methods are highly correlated. This indicates that the two methods agree on the choice of centroids to a large extent.
In conclusion, first, SCRAPT identified and removed large clusters early on in its execution, and therefore substantially speeds up the clustering process compared with other tools. Second, the improved efficiency of SCRAPT does not trade off the quality of clusters, as SCRAPT produces less fragmented clusters compared with other tools. Third, SCRAPT and DADA2 agree on the choice of centroids. For future work, we want to explore bias sampling procedures that increases the likelihood that large clusters found in subsamples selected in each iteration. We also plan to incorporate a support for full-length gene sequences in the future version of SCRAPT.